So this is my outline. I'm going to give you a brief introduction and talk about adaptive platform trials, and as Brian indicated, I-SPY 2 and GBM Agile. I do want to start with saying, you know, I'm not completely naive as regards neuroblastoma, just mostly naive. And Berry Consultants, and I worked with Christine Broglio and, of course, COG to help build this trial that Steve Du Bois tells me opened yesterday. So, and in neuroblastoma, and some very interesting adaptations, not precision medicine, but very interesting. And so that you can see that I'm not completely naive in the pediatric space. This was in last year's JAMA Oncology, and it's a point that I want to make. This is MRD negativity, and its role in drug development as not an endpoint in itself unless it predicts longer-term clinical outcome, event-free survival, overall survival, and that's what the paper was about. And in adaptive trials, we want to be able to build efficiency in, as everybody's been talking about, and to do that, you need early endpoints, you need early outcomes. And what we do is we model longitudinally. We look at the whole course of the disease. One of my pet peeves, when you read a paper in JCO or New England Journal or whatever it is, they tell you about overall survival. They tell you about progression-free survival. They tell you about response rate, but they don't tell you anything about the relationships among them. And that's the critical thing. We want to use the early endpoints to the extent to which they predict the longer-term endpoints. And we don't do this on the basis of, you know, some sort of external validation or whatever. We do it in the context of the trial itself, because that relationship may very well depend on treatment. And that's the bane of this business. And the difference between what the FDA calls a surrogate and a validated surrogate. If you ever hear the word validated surrogate, you got to look it up, because it ain't the English word. It's the regulatory word that they're talking about. And so this is an example. This is EFS, pediatric patients, 11,000 or so in 20 studies, that shows, you know, an enormous predictability of MRD negativity in this disease. And in fact, this was used recently by Amgen in getting approval in, an additional approval in ALL, citing this paper extensively for blinatumomab. So this is my one-picture definition of an adaptive trial. And the only difference between me and this pilot is I have a protocol. Of course, he might have had a protocol that said, if the propeller stops, get out and turn it. So there, as Brian indicated, there's all kinds of different adaptations, and I'll talk about this adaptive randomization, outcome adaptive randomization. This is my one-picture definition of a platform trial. So Gil talked about the eSmart. And this is Agile Inc. Yes, I like it. And the idea is that we evaluate drugs from various companies. The FDA has been solidly behind these kinds of efforts. They love the adaptive aspect. They love the platform aspect. Janet Woodcock has talked about this for years. She's the head of the Center for Drugs at the FDA. Improved utilization of adaptive Bayesian methods could help lower the failure rate of expensive phase 3 clinical trials. As you know, phase 3 trials is where the money goes down the tube. And what the FDA had noticed was that the big reason for these failed phase 3 trials is phase 2, phase 1, phase 2. and the horrible path to which you get to phase 3. And typically phase 3, historically, not only in oncology, but in all drugs, has been you roll the dice. You say, well, you know, we don't know if it's going to work or not. It looks positive from this. We've got an estimate in the phase 2 trial in response rate, and maybe that translates into overall survival. Let's roll the dice. And all too frequently, you know, these power calculations are fiction, and we end up with something closer to 50% success rate in phase 3. I should have given you some more. New England Journal, she touted I-SPY 2, the innovative groundbreaking trial in breast cancer to predict drug responsiveness based on the presence or absence of genetic and biological markers. More recently, said the FDA has to turn the clinical trial paradigm on its head to allow personalized therapies to get to the market faster. She doesn't know how to do it. I don't know how to do it. Nobody knows how to do it. We have to experiment with experiments. It's a bit ironic that the randomized clinical trial is like 70 years old. Some people have talked about it should be retired. It hasn't changed much in those 70 years. Name another technique in modern technology that hasn't changed in 70 years. All you have to do is take your thing out. Back when the randomized trial was first discovered or invented or initiated, Bradford Hill, these went around, and the only computers that were around would take a building this size to fit in it a fraction of the power that is contained in this thing. It's really quite amazing. The randomized trial, it's still, you know, we still kneel at the same altar to this idea. I kneel at the altar too, but I go down on only one knee. In the press launch of GBM Agile, Janet Woodcock presented and said, she ended her presentation with, "This is the future." And similarly, more recently, said the clinical trial system is broken, and one of the ways to fix it is master protocols, is what we call platform trials. And she wrote an article in New England Journal touting what she called master protocols, and I-SPY 2 is an example. Now, some of you may worry, in view of recent happenings, that Janet represents the old school, the older part, you know, she was appointed by a Democrat, et cetera. And maybe what happens in the new school, with the new commissioner coming in from the new administration, things are going to be different. So this is our new commissioner, and in his presentations and interrogation at his confirmation hearings, Kelly Cervic, writing for Science, said everything they asked him, he answered, we're going to do clinical trials from an adaptive perspective. If they said, well, you seem to be, you know, not too high on the usual high-level clinical trial, he said, we'll be adaptive. And she said repeatedly his zeal for adaptive trial designs. And in one instance, he was describing an answer to the question, what we're going to do is we'll have a phase 2 part, and then we'll morph into a bigger trial if it turns out that the data are good. And she said in this article, it sounds a lot like GBM Agile, and it is. So there are many platform trials, and you've heard some of them, in many diseases. This is not restricted to oncology. There's an Alzheimer's trial in Europe called EPAD that's specifically in the request for proposals for developing and running this trial. They said, we want to do I-SPY 2 in Alzheimer's disease, and they're doing it. So many cancers so far, I don't think neuroblastoma, but neuroblastoma is a wonderful example that would fit this paradigm. Ebola, antibiotics, and a number of other diseases. So in I-SPY 2, what's I-SPY 2? Brian has talked about it. The FDA, as I indicated, has touted it. During the trial, drugs are individually targeted to the biology of each woman's tumor by applying innovative trial design. Researchers then use data from one set of patients' treatments to decide treatment for future women who join the trial. This will help researchers learn more quickly which investigational drugs will be the most beneficial for women, certain biomarkers. So this is neoadjuvant breast cancer, and this is, you talk about turning the clinical trial paradigm on its head. How do we develop drugs in cancer? We first give it to metastatic patients. In breast cancer, no one starts out evaluating the drug in early breast cancer, non-metastatic breast cancer. So what does that mean? We've probably missed drugs because we gave it to people who had disease where the cat was out of the bag. And it may be, and then of course, we never get to the early setting. What we've done is turn that on its head, and we give investigational drugs to what the FDA worries about, potentially curable patients. And you'll see that we've had quite a number of successes. So it's neoadjuvant breast cancer. Primary endpoint is PathCR. There are 10 biomarker signatures, and I'll tell you what those are. We do adaptive randomization, which within each subset of the disease, and of which there are 8, 2 by 2 by 2, defined by hormone receptor status, HER2 status, and mammoprint, 70 gene genetic profile. We look at what is happening to patients with that disease, and we assign adaptively, because we've got a number of arms that we can assign, and we evaluate each of them within the category, and then we give with high probability. And I want to talk about one example of that. We do operating characteristics by simulation. Brian showed you a simulation. We do tens of thousands of those simulations within each scenario to evaluate how the trial is working and doing what we hoped it would do. The current status, we have 18 sites, 1,300 patients have been randomized, 14 experimental drugs, 10 different pharmaceutical companies, and these are some of the drugs. I want to focus on the longitudinal model aspect, back like the MRD thing, only in this case for MRI. PathCR is assessed at surgery about 6 months after the randomization. We do MRI at 3 weeks with the investigational drug in addition to paclitaxel, and again at 12 weeks. And we predict how the patient is doing. So it's adaptive within the patient as well. 6 of the drugs that we've therapies that we've evaluated so far have graduated. And of those, the sample sizes, first of all, the biomarker subsets where they graduated were different in the different drugs because they were identified as being, having different, sometimes complementary, sometimes overlapping signatures. And these are the sample sizes. So when we're adaptive, like this guy flying the plane, when he gets to where he wants to go, he lands. When we see that we've got an answer, we stop. That stopping is based on a prediction. Not all of the patients have been through surgery, but we do this prediction based on the MRI. And when it's looking good, we stop accrual. And I'll give you an example of that as well. When I started this, I knew that there would be great advantages to having many drugs and a single control. Randomized control throughout in all of the subtypes. The randomization comparison is to different control arms, and will be, for example, in GBM Agile, if the in HER2-positive disease, we have to give anti-HER2 therapy. So initially, we started out with giving Herceptin, and now we're giving Herceptin plus pertuzumab as the control. So I knew that there was a great benefit to just having a single control comparing many arms to it, but I didn't realize the great benefit that many arms offer in terms of maybe even, Ryan mentioned, comparing experimental arms. We compare experimental arms. We don't tell anybody, but we do, and we use that information to help build something that we call a time machine, something where we look at the control effect over time, and the control effect, not all of the patients in the control arm are being assigned concurrently to all of the therapies. We have like 3 arms at a time in the trial. Arms come and go. But a patient who's back here in time is helping with the concurrent comparison to the arms that are in the trial now, because those arms overlapped with the control, of course, but also overlapped with each other and arms that were back in that previous patient. So we get to use all of the controls in a discounted fashion using this modeling. The FDA calls it contemporaneous controls. So this is the articles that Brian showed you, and he showed you this picture. This is the setup in I-SPY 2. They're fixed biomarkers. In GBM Agile, we have biomarkers that are associated with the therapies that are coming in, or we allow for those kinds of biomarkers. In I-SPY 2, we have these fixed 8 subtypes. And if you ask, well, how many ways can you graduate a therapy because it's being used on all of these subtypes, it could be good in any one of them and bad in any one of them. And so you could graduate 2 times 2 times 2, 256 possible combinations of those subtypes. That's too many. We focus on just 10. So we have 10 biomarker signatures. One is all of the disease. Another is hormone receptor positive, hormone receptor negative, HER2 positive, HER2 negative, mammaprint positive. This is called triple negative disease, which I'll mention again, and it's something very important in breast cancer. Triple negative is regarded as being a bad disease because endocrine therapy doesn't work, anti-HER2 therapy doesn't work. Luckily, some other therapies work, and we've identified them. So those are the 10. So Brian mentioned neratinib as one of these things in the New England Journal, and I put this in the New England Journal, but it's only in the supplementary material, but it's really critical and explains a couple of things. One is that this is about halfway through the trial. Neratinib was doing well in HER2 positive disease, not surprising. It's a pan-HER family inhibitor. And so it was being assigned preferentially. If you ask, well, what was the proportion of patients assigned to neratinib who were HER2 positive, it was more than twice the proportion in the control arm at the same time. So patients were being treated better. At this time that I'm showing here, the DMC said to me, Don, you know, this is a bad drug to give. And it's not doing too well in this HER2 negative, mammaprint negative set. Maybe we should consider dropping the arm. And I said, Bob, you don't have to. The algorithm has already dropped it. First of all, it started giving with lower probabilities, and when it got a sufficiently compelling answer, it zeroed it out. It didn't mean it couldn't come back later as we got more information on patients who, hadn't been to surgery yet. But it never did that. It stayed 0. Meanwhile, so it's 0 here. Meanwhile, it graduated in a different set of subtypes, in a different signature. Now, suppose this were to happen in a real trial. Excuse me for saying that. What some people regard to be as a real trial. A two-armed, a dumb trial. I should have said a two-armed trial. You'd have to stop the trial, amend it, say we're not going to assign these patients in this subtype, and then try to restart it. Meanwhile, it would have been possible that you couldn't restart it in view of the negative PR. But it's easy to do. I mean, we didn't need an amendment because it was in the protocol. Nobody knew about this except for the DMC. So another example, we had pembrolizumab in the trial, and we announced in last year's ASCO the pembro results in triple-negative disease, the top box on the right, and hormone receptor positive HER2 disease. And it's really unprecedented results. 60% an estimate compared to 20% in triple negative, and again, unprecedented in a very hard to, the lower right is a very difficult disease to elicit PCRs, as you can see. The control arm, which is paclitaxel followed by anthracycline and cyclophosphamide, is only 13%, and 34% on pembro. So this is the final answer. 69 patients total, 29 of them were in triple-negative disease. Pembro graduated in triple-negative disease before these results. Guess how many patients we had through surgery when the algorithm said, this is a good drug and it should graduate. It reached our criterion of 85% success in a future phase 3 trial when there was 1 patient through surgery. And you're saying, how in hell could that happen? And the answer is, I told you we had MRIs. We had 20 or so patients through the first 3 weeks and another 10 or so patients through the 12-week. We had MRIs that were showing the algorithm, and I didn't believe this myself. I went back to look at the MRI data, and they were awesome. The tumors were melting away under pembro. And the algorithm, the probability distribution associated with pembro was not this nice squeeze thing. It was quite wide, but it was still such that it was almost impossible that it would not be better than control in triple negative. And indeed, the next 12 patients, many of those that had, we in fact didn't stop accrual. I told the DMC, I said, we can't go to Merck and say, your drug is graduated with 1 patient through triple negative. So we waited an additional 6 weeks and got more information, and the next 12 patients, 11 of them were PCRs in triple negative. And one of the issues that is critical is, what does PCR mean? And this is our overall data on EFS, and you see that EFS and distant recurrence-free survival is really highly predictive. The hazard ratios in both cases are 0.2, so 80% reduction in the hazard of recurrence. So GBM Agile. What's new and transformative in phase 3 trial? And the answer is everything. This is, the FDA has really bent over backwards and giving us giving us everything we asked for. It's a seamless shift from the kind of phase 2 part to a small confirmatory trial, stage 2. There are many arms that enter and leave the trial. There's a common control arm. We compare all the controls with this time machine. It has never happened at the FDA that they've allowed to use in a formal pivotal trial, a non-concurrently randomized control. And here they're buying into using this time machine. It's astounding. We continually update. We do monthly analyses. We do adaptive randomization in the learn stage. We identify arms indications. So as Brian indicated, we identify whether it's just recurrent disease or if it's newly diagnosed, methylated, or newly diagnosed, unmethylated, or various combinations. The interpretation of type 1 error, when you've got biomarkers and you're trying to identify, what is the subtype that the arm benefits, there are all kinds of different errors. You could say, well, it's beneficial in recurrent disease, but it turns out that the truth is, and when we simulate, we know the truth is it's beneficial in all of the disease, so we made a mistake. Or we could say it's beneficial to all of the disease, but the truth is it's only beneficial in recurrent disease. So again, we made a mistake. The type 1 error that we propose and the FDA agreed with is it's only a type 1 error if you identify a subtype and it graduates and it does well and you present it to us as a positive study if none of those patients in that subtype benefit. So it's a very liberal definition of type 1 error. And longitudinal modeling is not in there yet, but will be, and predictive probability. So I just want to tell you about the subtypes. Here's all GBM, newly diagnosed, recurrent, newly diagnosed in 2 pieces. This is a signature, this is a signature, this is a signature, and all of newly diagnosed and all of the disease. So there are 5 signatures. And when we add biomarkers, a drug can come in with a biomarker, then there are, I'm going to ignore the B biomarker, just consider the A biomarker. Then there are additional subtypes, and I went too fast. There are additional subtypes and additional signatures. You can have a signature that's the biomarker positive. You could have a signature that's the biomarker positive recurrent, a signature that's the biomarker, I'm skipping over those others. So Brian showed you this wheel. I want to take you a little bit around the wheel. Patient enrolls, randomized. We update the patient outcome data. This is all happening continually. We update the longitudinal model. We calculate the probability that each arm is better than control in each signature. We make decisions for those arms that are still in stage 1 as to whether you stop for futility, whether you stop because you reach the maximum for stage 1, whether you continue into stage 1 or graduate and enter stage 2. So these stage 2 patients are being randomized at the same time as the stage 1 patients. The only difference is that stage 2 has a fixed randomization, whereas stage 1 has an adaptive randomization. We continue, and if accrual is permitting, the reason I say that is we don't want to just burden the thing and not be able to get any arms through in any reasonable period of time, like 2 years. If accrual permits, we add additional arms. We update the stage 1 probability, and determine a randomization probability for the next patient that comes in, depending on his or her subtype, and we keep going. So it's a continuous process. I-SPY 2 is 1,300 patients and continuing. It's a never-ending screening process. So we require simulations for all of these reasons, but I'll stop there. Thank you very much.
My name is Yanlin Liu. I'm from Taipei Medical University, Taiwan. I wonder, I have a question for Dr. Vaso that in the traditional two-stage Simon design, two-stage Simon design for phase 2 study, when we're not using the adaptive design, but the traditional two-stage designs, I mean, it seems that for each small basket, for each arm of study, you have set a early stopping rule for very low response rate, while also a positive rule for very high, like a 35% or more response rate. I wonder what would you do if the response rate tested with that small population, with that small sample, get like a 20%, get somewhere in the middle. So how would you do with the result? And I wonder how would you tell the company what to do next? Thank you.
Well, first of all, it's an academic trial.
Yeah, yeah.
Second, the goal is signal searching.
Oh, okay.
So it's not a trial, a pivotal trial, is signal searching. We do have first-in-child trial. We will generate phase 1 data. But what we very much want to do is to identify active treatment with signals based on the statistical design you just summarized, and then discuss the company, because it will not stop. It will say, yes, we move forward. Oh. So this is why it's complementary to what was discussed by the other two speakers. It's just the beginning. It doesn't summarize what we will be doing. But with this program, we give access to innovation to patients, we generate signals, and then when it is, and I can tell you 2 arms have been dropped, and 1 is moving forward, and clearly this arm will continue into a way to demonstrate, randomized or not randomized, the activity of the combination.
Okay, so if the response rate is not too high, but also not very too low, so the decision will be between the PI and the companies to determine how to interpret the signal. I mean,
well, we set up bars what we considered for each of the one trial that would be relevant, because we want to have clear signals. This is the point.
Okay.
Do the bars differ by biomarker?
No.
They're all the same. Thank you.
Tab Trinair from Sydney Kids. I just wanted to pick you up on a point about the presence of subclonal ALK mutation, which you're now seeing by deep sequencing at diagnosis, and knowing that there can be an expansion of the number of patients with ALK mutation at relapse. So where would you set the threshold for giving an ALK inhibitor to a patient at diagnosis with subclonal disease?
That's an important question. I can see Julie from here, as we've discussed this in the COG, and I've certainly thought a lot about this. I think we need to, take a step back and really understand in our preclinical models whether subclonal ALK mutations will lend themselves to ALK inhibition therapy, and what is the threshold. And that really is why we developed this ultra-deep sequencing platform and sequenced our PDX models to be able to test the hypothesis. But and it's really not been done. So I think that data will help to inform whether moving forward we should be doing not just Sanger sequencing, but really deep sequencing, identifying these patients, and then ultimately should they be randomized to ALK inhibition therapy, or should they all be getting ALK inhibition therapy? I think that's a very testable hypothesis.
Thank you.
Let me extend on that. We get that question a lot about, and even with people that are enthusiastic about the adaptive design, sometimes they're overly enthusiastic and they think like, oh, if we choose the wrong biomarker cutoff, then the model will find that, and then we can use a different cutoff and a different cutoff, and there's still multiple testing. issues, and we really have to have a kind of an a priori biomarker decision. But Don, what do you think there's any room for that, for if you don't have a good sense of what the right cutoff is, or what the, maybe there's a allelic fraction that you care about, building that into the trial, letting the trial data figure that out, or is how narrow does the a priori hypothesis have to be to build it into a trial?
So the standard thing, as Gil indicated, is you ask, what do I want to see? And in his example, you wanted to see quite a substantial benefit. And I think the question was, suppose you're in the middle there. Well, not. I would build in, and actually the Simon two-stage is adaptive in just a very narrow sense, but even the Simon two-stage builds in, if you're in the middle, the possibility of going bigger for the second stage. So can you, these are always in phase 2 trials, not for registration, so there's, although sometimes it turns out they are, but that you have some flexibility in what you do. You're looking at the data and you say, well, maybe I want to continue. In fact, I mentioned that in I-SPY 2, whereas there was an example in Pembro where it had graduated, it crossed the boundary, but we said, let's continue for another few weeks. So I'm in a way, I'm mumbling, not answering your question, but you can rethink things after you're into the trial. Multiplicity looms, but in the end, you have the evidence that you have, and you can make a decision accordingly.
So would you say that what characterizes adaptive design is flexibility?
That the what in that design?
Flexibility in
Is that
the
how you
way to
characterize?
run the trial?
Yeah.
Or is it a raw word that should not be listened?
So the best thing is what we try to do. We try to build a robot to run the trial. And we teach the robot as much as we can about what might happen and how to act. So it's completely perspective. It's complicated as all get out, but the robot knows what to do. That means you can calculate type 1 error. How do you do that? Well, you set the robot in motion and see what it does, simulating. And you count how many times it makes a bad move, and that tells you the type 1 error. And then you may have to go back and change the design in order to accommodate the regulators in terms of what type 1 error should be. And moreover, it's the simulations serve a wonderful purpose. What we like to do is present the simulations to investigators and say, now look at what the trial is doing, the design is doing. If you were there, you're not going to be there, but you can be there vicariously. If you were to see it doing this, would you like that? And sometimes they say, no, that's too aggressive, or that's too liberal. I wouldn't want to stop here. I need more information because of safety concerns or something. And so we modify the design accordingly. So you can be flexible prospectively. If you're flexible not prospectively, then you fall prey to, it's an exploratory analysis, and we did this. And I've certainly been guilty of doing that, but I go to confession for my guilt because I say in the report, I did the following thing that wasn't in the protocol.
Yeah. But prospectively flexible is fine with me.
Good.
You were mentioning the 70 years of the classical phase 3, and I think today is very important because for most of the people in the room, we have been taught. Taught, you say in English? Teach, taught, taught?
Indoctrinated.
Yeah, I'm French, so I
So
don't.
let Don teach
What
you
is a
English.
classical phase 3? And the interaction with a statistician is always something lively, but sometimes difficult. So you said there is a robot, but the robot is a tool to better interact and make decisions together with the physician to prospectively modify things if it is required.
Yep.
Okay.
Let's have Lisa.
So one of the problems in pediatric oncology, which is a wonderful problem, Dr. Berry, is that we cure a lot of our patients, but our patients have significant toxicity. And in this era of precision medicine, I think that eventually, or even now, we're beginning to understand risk factors, biomarkers for those toxicities, and reduction of therapy or change in therapy from what we currently do now to some way of using those biomarkers to change therapy but maintain cure rather than obtain cure is going to be what I think is a challenge for someone like yourself to think about in terms of adaptive designs. How do we make sure that we can continue to cure certain patients and at the same time eliminate or reduce their long-term toxicity and use biomarkers to help us do that?
So it's interesting you say that because I was on a call yesterday with one of these foundation medicine-like companies, and they were trying to build something in immunotherapy. And I said, you have to factor in toxicity. It's not just people don't want something that just says, this is good. If you have this biomarker subset, you're going to benefit from the drug. The benefit depends on what the penalty is in terms of toxicity. So what I do, and in fact I'm doing now in I-SPY 2, is building a clinical utility index that you in fact look at both things. You try to evaluate, and this is working closely with clinicians and patient advocates, you try to evaluate what are the negatives here and how do they counter the positives. We did something like that in a diabetes drug. This is all well known. It's the drug is called Trulicity. It's dulaglutide. The first trial that we did was adaptive. I mean, it's amazingly adaptive. We had 7 doses of the drug, and we evaluated the doses against 2 controls, an active control and placebo. And we built a clinical utility index that was based on the registration endpoint, which is A1C, but also weight loss, and 2 safety endpoints, heart rate and blood pressure. And we evaluated them in a clinical utility index, and we did adaptive randomization to try to address, what is the best doses to use. We were looking for 2 doses to move into a confirmatory phase 3 aspect. And the doses were chosen on the basis of this clinical utility index. And it chose not the highest dose. By the way, the highest dose gave the best result in terms of A1C and the best result in terms of weight loss. But it didn't choose the highest dose because of safety issues. And there, by the way, when I design a clinical trial that's adaptive, I like discussing with the data monitoring committee. They know they can't change the design, but they say things like, as this particular data monitoring committee said, I know we can't redesign the trial, I know we can't design the trial, but if we were able to do that, we would do exactly what the algorithm is doing.
And the next step will be as well to get the opinion of the patients for the clinical utility index, no?
Yeah, absolutely. And
That's hard to do with children.
We work with patients.
But we were discussing breast cancer. This is
No, but it's hard to do with children.
But
What
I
a
would think that the
parent will
parents
choose
as
for
well.
their child. I mean, it's just hard to do. That's all. What it means to be to grow up hard of hearing because of
Yeah,
the chemotherapy that you got? And could you
it's
design a
really
trial
hard.
where, I imagine a clinical trial where we randomize our high-dose platinum to some other thing or some odor protection that gives you a higher rate of relapse potentially, but less hearing loss. Well, that is hard to imagine spending 5 years answering that question in a traditional phase 3 trial. But I've learned a lot this afternoon about other ways that we might look at that, including a utility index, and then figuring out what that utility is by large samples of patients who've actually survived disease with hearing loss or not, as well as parents and maybe patients. I don't know.
The problem is that different patients have, not it's a problem, it's a reality, different patients have different utilities.
That's right.
So the ideal would be for the physician to sit down with the patient and have,
That's
an
right.
hour or two session where you say, well, suppose this, how do you value losing your hair as compared with, vomiting or going to the bathroom for
Right.
too much.
But for us,
it'll
it's
vary.
issues like the value of premature ovarian failure 20 years later versus hearing loss versus
So-
short stature. It's
Yeah.
just,
So in practice, you sort of try to get a consensus
Yeah.
of what the utilities might be, but you still have that conversation, maybe not a 2-hour
That's right.
conversation, but you still have that conversation.
Just a follow-up of that. So in this clinical utility model, you make the assumption that everyone has the same risk for the toxicity, and which we know may not, that may not be
So
true.
do you
because
mean the
may-
same risk?
So
You
make
mean
an
like,
assumption,
for example?
so say someone has a genetic predisposition to a toxicity. If you use your clinical utility, you may say, well, we're going to choose this age, this dose. But really, if you looked at it in a different population, you might have chosen a different dose.
Well, I mean, you could
So
think about the
DPD
toxicity.
deficiency, for example.
Yeah.
Yeah, you could think about the toxicity the same way as you think about an endpoint, right? You could
It
have
would
a
be
biomarker for that.
for a toxicity. That's
Yeah,
what
yeah.
I'm thinking. Yeah.
And then you could have a longitudinal model for the toxicity too. I mean, that would be great if you could predict the toxicity, a long-term toxicity, years before based on some kind of bio. I mean, I just don't think people think. I mean, maybe having more of these models that include toxicity would necessitate some of those ideas having to do that kind of thing.
and then, because we've been studying long-term survivors of childhood cancer for a long time, and now they're adults, we actually have utility data from them about their lives now based upon specific toxicity that could be put into the model.
Yes, I mean,
you don't like
that's
that.
a good thing to consider, but it's really, I'm reminded, I read
Quality
somewhere where a physician said that he was seeing a patient from one of his colleagues, and he'd never seen her before, and she had a radical mastectomy back in the, in the early days, where they, went to the
Yeah.
chest wall
It's not
and
the
removed
same as-
ribs,
yes.
and he said she went through hell. I mean, it's amazing what she. And so she came into his office, and he said to her, "Boy, that must have been a bad experience." And she said, "No, it wasn't too bad."
Yeah. No, this is more about the quality of current life, not so much about recalling. But yes, I understand what you're saying.
Thank you. So I think that's all the time we have today, but I'm sure everyone's available afterwards to discuss more. But thanks very much.